Welcome to my poster number P052.01 entitled Pharmacology of Transient Receptor Potential Cation or TRP Channels using Different Activation Stimuli. My name is Ali and I work for Transient Technologies, the manufacturers of the automated patch clamp instruments used in this study. With this poster, I would like to give you a quick introduction into automated patch clamp and give you examples of different TRP channels used on automated patch clamp devices with varying throughput. In case you're not so familiar with automated patch clamp, I will start off by explaining the difference between automated and conventional patch clamp. In conventional patch clamp, a small glass pipette with a tip diameter of approximately 1 micrometer is brought to the cell surface and a tight seal in the order of 1 gigaohm or more is made between the cell membrane and the glass of the pipette tip. Following this, single channel recordings of ion channels trapped within the patch of membrane stuck in the hole can be recorded, or the patch of membrane can be broken to access ion channel activity occurring across the whole of the cell membrane. With automated or planar patch clamp, instead of a pipette, we have a planar glass layer with a small hole in the middle of it, also about 1 micrometer in diameter. The cells in suspension are pipetted onto the top of the chip, and suction is used to attract a cell to the hole to make a good seal and to break into the whole cell mode. The systems are easy to use and can be scaled up to record from multiple cells simultaneously. In the experiments described here, we used either the Porter patch, patch liner, or SynchroPatch 384i for the measurements. The Porter patch is a semi-automated system recording from a single cell at a time. The perfusion system is temperature controlled, which can be heated as well as cooled. In the experiment shown on the lower left panel of the poster, solution was cooled to 10 degrees C and used to activate TRPM8 expressed in CHO cells. In panels A and B, solution was first cooled to 10 degrees C to activate TRPM8 and then heated to 35 degrees C, which reversed the activation. Following this, TRPM8 was activated a second time using cooled solution at 10 degrees C. In the next experiment, shown in panels C and D, TRPM8 was activated by cooled solution and then blocked by either capsaicin or heated solution. Capsaicin could be washed out and the current recovered. A concentration response curve was performed by adding increasing concentrations of capsaicin at 10 degrees C, which is shown in panel E. The curve was fit with a Hill equation revealing an IC50 of 12.9 micromolar, in good agreement with literature values. In the next experiment shown on the top middle panel of the poster, we recorded TRPA1 expressed in CHO cells on a high throughput automated patch clamp device, the SynchroPatch 384i, which records from up to 384 wells at a time. In these experiments, we used multi-hole chips with 4 holes per well, and used the perforated patch mode. TRPA1 was activated by carvacrol, shown in panels A and B, in a concentration-dependent manner with an EC50 of 308 micromolar for an average of 355 wells. Panels C and D show block of TRPA1 by A967079 and AMG 0902 respectively. TRPA1 was first activated 3 times by 300 micromolar carvacrol to check that the current was stable, and then carvacrol was co-applied with the inhibitor. The IC50 for A967079 was 12.4 nanomolar for an average of 320 wells, and for AMG 0902 was 48 nanomolar for an average of 349 wells, both in good agreement with literature values. In the next set of experiments shown in the bottom middle panel of the poster, the patch liner was used to record TRPV4. The patch liner is a fully automated patch clamp system for recording up to 8 cells simultaneously. TRPV4 was activated by the ligand GSK 1016790A, a potent and specific activator of TRPV4, and blocked by GSK 2193874, a specific blocker of TRPV4. TRPV4 could also be moderately activated by heated solution. To do these experiments, the solution was heated inside the pipette of the patch clamp robot to the required temperature, in this case 45 degrees C, and then rapidly applied to the cell. In 34 out of 56 cells tested, we could observe a moderate activation of TRPV4 by heated solution at 45 degrees C. Finally, we recorded TRPV1 and TRPV3 using either ligand, shown on the top right panel of the poster, or using heated solution, shown in the middle right panel. Capsaicin was used to activate TRPV1, and this was blocked by capsaicin. 2-APB was used to activate TRPV3, and this was blocked by ruthenium red. Both TRPV1 and TRPV3 could be activated by heated solution at 45 degrees C. TRPV3 activation sensitized to repeated activation by heated solution. With repeated application, current size increased. This has been reported in the literature and is a phenomenon we have seen previously with TRPV3. In conclusion, we have recorded several different TRP channels using different stimuli. TRPM8 was activated by cooled solution and blocked by capsaicin. TRPA1 was activated by carvacrol and blocked by A967079 and AMG 0902. TRPV4 was activated by GSK 10167990A or heated solution and blocked by GSK 2193874. TRPV1 was activated by capsaicin or heated solution and blocked by capsaicin, and TRPV3 was activated by 2-APB or heated solution. Thanks very much for visiting my poster. If you have any questions, please don't hesitate to ask me using the chat function at the SfN Connectome meeting, or can contact me anytime by email on ali@nanion.de. Visit the Nanion website at www.nanion.de or find me on LinkedIn. I look forward to hearing from you and wish you all the best for 2021. Enjoy the SfN Connectome meeting.